Okay, so as promised, here we are going to take a look at the DNA marker analysis process, so to speak, just one more time. I wanted to go over the two slides that we reviewed today in class. Before I do that, I want to just point out that traditionally it has been expensive for people to have DNA cycle sequencing done on their DNA. So not too many folks have paid the extra, or has their insurance been able to pay for a DNA test that involved sequencing, just so that someone could identify whether they should be diagnosed with a mutation that may cause a disease or not. Less medically necessary. I wouldn't think insurance would cover it. So anyway, nowadays, scientists and medical doctors are suggesting a different route, which is the marker analysis tool. And basically what it allows you to do is obtain an image of someone's DNA fragments in a region that is adjacent to a particular gene of interest. So speaking of breast cancer, we're interested in the BRCA1 and 2 mutations. In our case, we've decided that the BRCA2 mutation is what's present in the Smith family. The BRCA genes are tumor suppressor genes, so when they are mutated, they no longer tumor suppress, and that's the problem. So here we have an image of where the BRCA2 gene is located, and that's shown to you here in kind of this bluish color that you see. And then in addition to that, we also have the red section, and what that red section actually is this adjacent area. And the adjacent area is known as a marker because it's basically correlated to the gene of interest, and therefore we can look at that marker and identify if someone has a particular mutation or not. And here's how that works. Basically, what we look for are what are known as STRs. STRs are typically located adjacent to an area that may become mutated. So let's just say, for hypothetical, that man 1, man 2, and man 3 are having their BRCA2 genes checked using this process. And so what we've learned is that man 1 will later show that he has BRCA2. And right here, this single nucleotide polymorphism, in other words, this nucleotide, is changed as from what it should be, which is typically an adenine or an A. So in this case, man number 1 has a T, but we may not know that yet. So what we would do is take another area of the genome adjacent to the BRCA2 gene, and we would look for a short tandem repeat. And we would count how many repeats exist in this individual's genome. So we're looking specifically for a repeat here of CTA, and you can see that on man number 1 he has 5 repeats. Now man number 2 has 6 repeats, and man number 3 has 7. So clearly they have more repeats than this individual up here. You'll notice as well that the number 5 of the repeats happens to correlate with a mutation in that particular SNP. Man 2 and man 3 don't actually have that mutation. But again, we don't know that because we haven't sequenced their individual DNA nucleotides. But because of the number of repetitions that differ from each man, if we were to snip out or cut out this particular region of the genome here, 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 and so on, I don't know how long the gene actually is, so it could be pretty long, and run them through a gel electrophoresis, what we would hopefully notice is that this one is going to be the shortest because of the 5 repeats, as opposed to man 2's fragments are going to be longer. There's 6 repeats there. Man 3 is going to be even longer than that, and that's 7 repeats. Now in a gel, and again, I wish I had a picture of this to show you. Yeah, it's not real clear here. The larger pieces of DNA are going to move a little bit slower through the gel. So I might see man 3's fragments up near the top of the gel. Man 2's might go a little bit lower, and then man 3 is going to be, even lower than that. And so with that in mind, what we can also do is take a person that we know to have been diagnosed with the BRCA2 mutation, probably someone who's had DNA cycle sequencing done on their strand. And again, you might do that if someone inquires as to the cause of their breast cancer. So they want to find out if it's genetically linked or if it came about on its own from some other means. Anyway, they might actually do the DNA cycle sequencing in that case because insurance in that case might actually pay for it. As you know, we've talked before, the process isn't that expensive these days, but it used to be thousands and thousands of dollars, and people just weren't paying that. So long story short, if we put a fourth or fifth person's DNA profile in the same manner, in the same gel, what we might find then is that man one has the exact same length fragments in the gel. And then if it looks the same as those people with a diagnosed BRCA2 mutation, that leads us to recognize that man one has the marker that hints at him also having the same mutation. And the reason for that, again, comes back to the correlation between this number of repeats and that letter right there. So essentially what scientists recognized in order to develop this was that these repetitions were common amongst people who had the particular mutation we're talking about here. Whereas any other number of repetitions, a 6 or 7, I don't know how high it goes, did not have the mutation. Maybe they have to have just 5, or maybe it was 5 and 4, or something along those lines. This is kind of an oversimplified example here. But I hope that helps. I told you I would come up with a metaphor. The only one that comes to mind is something that we should do repetitively in order to earn a good grade in a school course. So let's say that studying is what helps to actually obtain the better grade. The grade is essentially this right here. It would be telling us the SNP. Okay, what version do they have? Do they have an A? Do they have a B? Do they have a C? And the more repetitions or studying sessions that someone were to do is going to essentially correlate with the letter that arrives here. So if someone were to study content 5 times, let's say, we'll use the same examples as are here, they would end up earning maybe a C in an assessment. Oops. If they had 6 repeats, maybe they earn a B, and if they have 7 repeats, maybe that's the equivalent of an A. And I'm not talking genetics here, obviously. I'm talking about something metaphorical that's in a very different context. But long story short, let's say we could quantify, which you could, the number of hours that someone has had a study session. You could put that on a graph and see a correlation between the grade earned on an assessment and the number of repeats. That is more or less the same thing as what we're talking about here, but in a very different context. So I could probably look at the graph and see, based on the fact that this student earned a B and anyone else who earned Bs studied 6 times, this student probably studied 6 times. Obviously, that's an oversimplified metaphor, but that's kind of the same thing as what we're talking about here. Okay, now try and explain that to someone else, and if you can explain it well, it probably means that you understand the process all right. I'm going to wish you best of luck on the assessment tomorrow, and that should be it. Okay, have a good night. I hope you study well. Thanks. Bye.